Ever wondered how AI can be used in cancer research? Then come along and find out. Today's guests are Professor István Csabai, physicist, and his PhD student, Oz Kilim, also a physicist, from the Department of Physics of Complex Systems. Hello.
Hi.
Hello, thank you for having us here, and hello for the online viewers also.
Thank you for coming. Your team that included researchers from the Eötvös Loránd University, Harvard Medical School, the Danish Cancer Institute, and the Semmelweis University has developed an innovative approach to predict how patients with high-grade serous ovarian cancer respond to chemotherapy. Let's start at the beginning. What's high-grade serous ovarian cancer and what makes it especially hard to treat?
Yeah, so this is a certain kind of type of cancer within all the cancers of ovarian cancer. And many cancers, not just high-grade serous ovarian cancer, have kind of often find ways to avoid treatment through kind of evolution. And it makes it quite difficult to treat these cancers with platinum, which is kind of the main way of using implementing chemotherapy now for patients. So some patients are doing very well, some are not responding on their first treatment and need to go for further treatments down the line. Some are getting worse. So I think this is a big open problem now that people want to work on to be able to know which patients will kind of respond better and less well ahead of time, so you don't have to give patients a therapy that would be kind of disadvantageous to them.
How do doctors typically diagnose this type of cancer, and how do they decide how to treat them? Because there are a lot of treatments.
Yeah, so I think firstly there's a whole team that will come together. There will be pathologists. First they'll be looking maybe through imaging, through MRI or through ultrasound and so on. And then clinicians may take a biopsy and look at the tumor kind of under the microscope and see the cells and decide how aggressive this tissue is, this tumor. And from that, they will decide on what the treatment should be. And I think, as far as I'm aware, most of the patients are going for chemotherapy as the first line treatment. And there's more move also towards doing chemotherapy before doing a surgery to remove the tumor. But now often people are having the tumor kind of taken out with surgery called debulking. And then after they'll take chemotherapy to kind of hopefully clear out kind of any remaining tumor that's left. I don't know if István has some other...
Yeah. So actually, just in general, so this is right now the typical way of how to diagnose and treat cancer. And now we are moving towards the personalized area, because so there is no one-fit-all method. So right now we are using something that is best in general for all patients, but there are differences between patients. So for some patient it is working, for some patient it is not. And now to make this diagnosis that for this specific patient, which one of the choices would be the better, that's the real question. That's personalized treatment. And this study is trying to find something to decide whether for the specific patient, the best method that is good for most people is good for that specific person or not.
There is this other type of data from the tumor cells, the proteins inside of it. How can it be used?
Something that kicked off this study for us was a recent publication in Cell, which was using this kind of measurement, this proteomics, which is kind of a very granular way of measuring how many proteins of which types are within a tumor. And this seemed to carry quite a lot of prognostic signal or kind of information to be able to predict if a tumor will respond or not respond to chemotherapy. And these numbers are pretty good. And basically, we wanted to see if we can improve these values by integrating other sources of information. But this seemed like a good baseline. And in the clinic, there are baselines based on genetics and specific mechanisms known to be related to platinum response or non-response. So this is the homologous recombination deficiency kind of status of a patient. And these are kind of known biologically established mechanisms of resistance that people can look at and make decisions on, which have a certain prognostic value and are used in the clinic. But the proteomics is something kind of quite new and not really used in the clinic yet for this specific cancer type. But they showed to be very promising. And so we thought this would be the direction we would kind of go in. Yeah.
So you combine this with pictures of tissue samples,
Yeah,
right?
yeah. So this is this other modality of this the histopathology, and this whole area of digital pathology is really kind of blowing up right now because of the advance of computer vision, where you can kind of compress very high dimensional image data into more low dimensional form that you could do machine learning with. This is kind of a knock-on effect from back in 2012 or 2012, like ImageNet being able to be processed by by neural networks, and this has kind of had a domino effect onto all different fields in computer vision. So this is something that's there's a lot of developments in that side as well. Mm-hmm. So we thought it would be kind of worthwhile to try and merge these two emerging sources of signal together to build models that hopefully can be used in the clinic.
What was the biggest challenge when you tried to combine these two types of data? Because one is an image, another one is something I don't know exactly.
Yeah. Yeah, so the proteomics is like you can think of it like as just a vector, mm-hmm. and the image kind of becomes a vector, and you can either just kind of concatenate them or just join them together, or you can join them at kind of earlier stages. So there's a lot of technical challenges in, I guess, in both. One is correcting kind of for batch effects within the proteomics data. In the digital pathology side, there's kind of the fact that these images are huge images and you need to slice them up mm-hmm. and patch them into smaller kind of manageable sized images for neural networks to kind of compress. So there's lots of different ways to combine the modalities, and we played around with a lot of different kind of methods. And yeah, I think these are the challenge, I guess, is that there's no one clear-cut answer. And different methods don't necessarily, because they work in one place, doesn't mean they'll necessarily carry over. So it's a bit of a like free-for-all of which method to use, and often you have to just try it on your specific data set. Yeah.
How big was your data set to achieve meaningful results?
So we're in kind of the low hundreds in terms of
mm-hmm.
Patients in the training set and the validation sets. And we found that this is kind of a reasonable place to get some kind of competitive results. I would imagine they'll improve as more data is available, but this is one of the bottlenecks, I guess, in this field, is because medical data is very protected, as it should be, it makes it quite difficult to make very big cohorts of patients and build models on very large data sets. So I think it's promising that on the low hundreds, you can get kind of some signal out.
Yeah, but maybe extend a little bit this. So we have used a model mm-hmm. that was already pre-trained for the images. And this is a big thing, these so-called foundation models that they were trained on hundreds of thousands of images, actually. So like similar to the large language models, that they are trained on the whole internet, they read everything on the internet, and they have a basic knowledge, which is very smart. And we have those similar models that were actually trained on histopathological images. Mm-hmm. A single image is sometimes several 100K by several hundred K. So these are gigapixel or sometimes terapixel images. So a single image is that huge. And there were hundreds of thousands of such images on which it was trained. Because as Oz mentioned, a critical point here is that if you have this device or that device, this microscope, that microscope, and in the lab the staining where they dye or paint the images actually, there may be differences because of temperature, concentration of reagents, and so on and so on. So everything is a little bit different. And the model has to learn all these differences, and they have to learn that these are not important for the specific question. And actually, with these models, which are, I think this is just the one thing to say beyond the concrete topic, that this is really, really good, that all these models are shared. So I think this is a big success, not just for science, but in a sense, if I may say big words, for the human race, that we are sharing this. So there are these basic models that were first trained on ImageNet, cats and dogs, whatever, and these models were shared, the method, how it is done. And then we trained more and more larger and larger images, and now colleagues at the US trained it on hundreds of thousands of histopathological images, and they shared the model. And we can just download from GitHub the model and use it for this next step. And actually that knowledge that is compressed into the models, that helped our study, and we could make it on a relatively small data set, just 100 patients, because otherwise, if we would have to start it from scratch and train the whole model, we would get nowhere.
So in reality, it's a more bigger collaboration. And
definitely, yeah.
The whole world is working on it.
Yeah, and I think everyone is building on top of kind of each other's work, and yeah, this was a big step forward, these foundation models for this specific area. Although cats and dogs have got us pretty far, surprisingly. They're pretty good, even the ImageNet pre-trained vision models. But yeah, the next leg up was pre-training on histopathology in unsupervised, self-supervised way without labels. So there's people who've been working on self-supervised learning, and people have been working on taking that into histopathology, and all these things together kind of have had great effects, I guess.
Yes, and also we now shared our model, and other people can build upon that. And actually we need this because, as we see, cancer is really complex. So really, really complex, and we need to work together and share everything to get to somewhere.
How big is the improvement of your model compared to using only just detailed tissue images or the protein measurements from tumors?
Yeah, so I think our main thing that was surprising is that by themselves, both of them are not very useful. Mm-hmm. They're not really at the kind of clinical levels, kind of accuracy that someone would actually take this on and use it in hospital. But when you combine them, you get kind of this synergistic effect where kind of they are unlocking the signal in each other. It's kind of a bit of an airy way of saying it, but they're kind of they're giving each other context that allows for better performance overall. So I like to think of this as there's a good picture, but I will try and describe the picture, which István has actually sent me, which is a spiral where there are arms of the spiral, and the spiral is in like a 2D space. So it's like a picture of spirals with the arms of the spirals, and each arm could be a class, like responders or non-responders to chemotherapy, and each axis could be one of the modalities, like the histopathology and some omics or the proteomics. And by themselves, you can't distinguish which arm you're in very well, depending on where you are on each axis individually. But when you have information from both axes, you suddenly can see which arm you're on. This is probably really bad to describe just in words, and a picture would make it much more clear. But I think there is some, I guess, theoretical grounding in why you get such positive results from using multimodal data.
Could you imagine this method being used in clinics?
I hope that eventually it will. It won't be our model specifically that was trained on our computer now, but I think it's about showing people that it's possible and these kind of synergistic signals exist and people should take them on and push them further. So I hope they will be used. And I think people will include more modalities. And there's a bit of a kind of cost and ease to quality, to interpretability, kind of weigh up. Not that you can necessarily weigh up like three things, but as things become more complex and more modalities are involved in models, the interpretability can get it can become interesting in a way, but it can also become a little bit scary for maybe the legal side or the kind of FDA kind of side of things. But I think that's why people will now go for more simple models that have some kind of clear biological interpretability. And these kind of deep learning, high dimensional data models are a little bit slower coming forward, but they're definitely going to go forward, and I think people will adopt them. It's
so
a
the
very long
doctors
answer.
can look at the results and use it as an extra information, let's say.
Yeah, I think that would be the goal. But I think it's still a little bit of a while off till it's really operating on patients, because these are very important decisions.
Yes.
And it's a little bit scary if just some weights, some kind of numbers are just kind of multiplying and they just decide, like chemotherapy, not chemotherapy.
Yes, but they are already deciding based on the tissue samples. Yeah. So it's just if you think about it as an extra information, then I think it's a huge improvement.
Yeah.
You also found that studying the surroundings of the tumor could offer valuable information. Could you tell me more about this?
Yeah, so this whole like tumor microenvironment topic is a known thing, that the environment of tumor cells is strongly affecting the behavior. So this has been studied with doing sequencing, kind of spatially resolved sequencing, so where you can sequence tumors from all different parts of the tumor and kind of get a bigger picture. And it becomes clear that it's much more complex than just sampling one part of the tumor or mashing the tumor up and taking an average kind of view of things. This is like, people often say that this is like having a smoothie versus having the fruits themselves. You kind of get a lot more when you look at the fruits. So I think that what we saw is that because you can get these kind of attention maps from the model, where the model is looking at on the image in a spatially resolved way, we could see that it wasn't just the tumor cells that were gathering attention by the model, but also the kind of nearby stroma or different parts of the tumor that people know are important, but this further kind of emphasizes that they might be directly important in the signal they carry for this task.
Actually, this is, I think, quite an interesting and a little bit surprising part. So actually, there were these two input modalities, like proteomics, which was just a single measurement on the whole tissue. So it was not spatially resolved. And there was the image, which was kind of black and white. So actually, it's pink and purple, but actually it's kind of black and white. So just a simple image, not colored. And actually, the model could connect the two information in a way that then it colored the image by the activity of genes. Okay, that's really surprising because doctors, pathologists cannot do that. If I give them the original image and say that please, and this is the list of genes that are active in proteomics, and please color this image like in a color book, please color the image by the gene activities or gene pathway activities. They would say that this is not possible. And the model can do that. And it is a little bit surprising, but on the other hand, we have this analogy with cats and dogs again, that if you train a lot of images taken from Instagram or whatever social platform, and you have also the text below the image, but no one has circled around cats and dogs. But if the model sees enough number of images with the caption that there is the dog walking in the forest or whatever, then it will recognize that in pictures where there are the dog is, and you can see the word dog in the caption, then there is this correlation, and at the end it may find, and this is this attention map, this finds the attention focus on the image where the furry little creature is. So, and this is the analogous way, but no one looks at big lists of proteomics, gene activities, and histopathology images to learn that. And the model, because it is fast and can handle very large data sets, it could learn that in a very similar way as we learn from words what are in the pictures.
How can all of these lead to more personalized treatment plans for ovarian cancer patients?
So I think the big thing and the big focus, which was really emphasized to us from our colleagues who are more on the clinical side, and they feel what would be important and useful most, which I think is really important in these collaborative settings, to really listen to what the other sides feel would give them the most value. We found that predicting this first-line chemotherapy is kind of a very basic and important thing to kind of work on, because you have a therapy that's working already, and it's approved and it's in the clinic, and if you can just give it to the right people, you can make a big impact. And so this is focused on personalized therapy, not from the drug discovery or the therapy discovery kind of angle, but just taking what we have already, that we already know works for some people, and making it and selecting the people better. Mm-hmm. So I guess it's more about selecting the people for the therapy than the new therapy. And the second thing, just to add to that, is that when you can predict if a patient will respond or not respond to chemotherapy, if they don't respond, you can put them on a track for a clinical trial where they have a higher probability of responding, and they don't have to have all the terrible side effects that they might have. So I think this is a step in the direction of personalized therapy, but it's a huge field and a lot of people are working in this area. So I don't want it too much sound too high.
Yeah, so I just want to highlight that was mentioned the collaboration between people. So there were people from Semmelweis University, Medical University, and from Harvard, Danish Cancer Research Institute. So I think this cannot be done in another way. So there are people who are more expert in machine learning, data science, people, doctors, and we need to get together all. And as you see, Oz is a physics PhD student, but he's talking almost like a medical doctor. So we learn from each other actually, but definitely separate researchers as a separate expertise. And I think this is also very nice, but a bit challenging to get together, learn the other's language, see the focuses, how they are different, see what is possible and what is not possible. What seems to be easy for me, it's not necessarily easy for them, and so on and so on. So it's really, really fun and kind of challenging to do this.
Yeah, I wanted to ask you how was working in this big collaboration with researchers from other fields?
It was really nice. I think we had a really good kind of collaboration on this project. We had experts in pathology. We had pathologists, geneticists, on kind of general oncology kind of background, collaborations, and physicists and machine learning people. And I think it was a really nice kind of experience to see everyone come together and navigate this collaborative landscape. I've in the past tried to decide what problems I should work on, but I find that going to people who really know what would have the most impact can kind of offer a lot. So I think this is a good kind of direction, I think, is to work in a collaborative setting and not just by yourself, deciding what problem you think is important necessarily.
And you are here in Hungary with the Stipendium Hungaricum scholarship.
Yeah.
How did you decide to come here to Hungary?
Yeah, so this is interesting. So my wife is Hungarian, and I was looking around at whose lab I should join, and I wrote to Professor István, and he wrote back, which was very, very nice. And I didn't know at the time about kind of this group and this field, and I knew I wanted to get involved in this area, and I think it just worked out very well that I could join the group and be involved, and so I'm very grateful for how it's all really gone, and I've had a great time in Hungary and continue to do so.
What was your background before doing this?
So I trained as a chemist, which is really a bit kind of random and off. But then I realized I was very interested in this whole machine learning area, and I was interested in cancer research as well. So I found that this happened to be kind of a nice niche little thing. It's not that niche because a lot of people work on it, as I said, but it's a nice area, a kind of pocket of research. Yeah.
And you have this big collaboration with your people, István.
Yes. Actually, I am really lucky that I have such talented students like Oz and many others also. And I think that's also good that we have this team, we have this local group here, and some of them are working on astronomy, other applications of machine learning. So the technical side, they are all the same. So we have, for example, images or various scientific data sources, and we try to move forward that specific discipline. And they can interact with each other. Sometimes someone plans that I will do this and that and hear something from the other side, and it moves that faster forward. And also, I think it's nice for students also to be involved in such projects where you really see the goal, where it's not just a theoretical or methodological development of this thing, but that there is something is pulling towards some goal, and to collaborate with real doctors, real people, see some real effect, as was very cautiously said, that we do not know whether it will be used or not. This is definitely a small step, but at least we may think that we work something towards some greater goal to cure cancer in the long term, and I think this is for most students and for myself also. It's very important to do something that we think that can be really important and that can have some real good effects.
And as you said, you share your results with the whole world, and they can build up on that. So I think it's really making a huge impact, even if you don't notice it right away. What are your plans for the future?
I think in this area of kind of research, I think a lot is happening. So it's a very exciting time. I think a lot of new ways of measuring cancers emerging, and I think everyone understands that publishing larger data sets and so on is important. So I hope to continue in this area with more high dimensional data and more kind of granular data, and hope to continue to work on the same kind of tasks, because as István said, that it's very important, and it can have some kind of real effect in the world. And we're working on a lot of random things as well. Professor Czabai is exceptionally open-minded, and kind of is dabbling in a lot of different things. And I think it's really great because sometimes we can combine random ideas and so on. So we're always brainstorming things.
We have crazy projects also.
Yeah.
Not just these nice official projects.
Yeah.
That's really good. And also, I just wanted to add that through these collaborations, we have new opportunities. So Oz is soon finishing his PhD, and he will decide, or it will turn out whether he stays with us or he can work together with one of our collaborations in Boston or Copenhagen or wherever. And we have just won together, actually based on this study, a grant from Microsoft and the Ovarian Cancer Association, which will provide some funding for future work on this topic.
Thank you for being here, and I wish you all the best in your research, and thank you for coming.
Thanks very much for having us.
Thank you very much.